Okay, let's talk about free radicals now. Now, a free radical is a chemical species that has an unpaired electron within the outer orbit, and that unpaired electron makes that compound really highly unstable, and it could be damaging to the cell. Now, free radicals, they can occur physiologically, or we could get them from a pathology. Now, in the physiological aspect of free radical formation, uh, you all remember something that is called oxidative phosphorylation. Now, in order to generate ATP, we need something that is called an electron transport chain. And that electron transport chain consists of electron transporters such as cytochromes, especially cytochrome C. Now, cytochrome C basically transfers electrons into to oxygen, and basically we generate something that is called a proton pump, and we activate the ATP pump, and we generate ATP. Now, the thing is, sometimes partial reduction within that process, partial reduction of O2, can lead to the formation of free radicals. Now, for example, we normally, the O2 gets 4 electrons and it is transferred into H2O. Now, however, partial reductions, for example, let's say that O2 only gets 1 electron, so it will turn into superoxide. Okay, let's say it gets 2 electrons, it will turn into hydrogen peroxide. Let's say it gets 3 electrons, it will turn into hydroxyl. And actually, hydroxyl is the most damaging of all the free radicals. And now, those free radicals are damaging to the cell in 3 ways we will talk about in a minute. But when we say that they form naturally, they are physiological, now that doesn't mean that they are harmful, because we have a couple of clearance systems within our bodies that help us get rid of all those free radicals. So you should understand that the formation to clearance ratio of the free radical is important in understanding why the free radical is injurious. So that accumulation of free radicals can cause cellular injury, and a defect in the clearance of free radicals can also cause cellular injury. Now, we said that the free radicals, when they form, let's forget about the pathology for a minute. Now, we said that the free radicals, when they form, we can actually basically clear them out of the cell. Now, for example, let's see the superoxide. Now, the superoxide free radical gets cleared by converting into hydrogen peroxide by some enzyme that is called a superoxide dismutase. Okay, so this is number 1. This is the first enzyme of clearance of free radicals. So superoxide dismutase turns superoxide into hydrogen peroxide. Now, hydrogen peroxide gets into something that is called a glutathione circle, or a glutathione by a glutathione peroxidase enzyme, or a catalase enzyme, and those are enzymes number 2 and 3, and they convert it basically into water. In the glutathione system, let us draw something here. This is H2O2. Okay, and we have the glutathione, the GSH. Okay, by glutathione peroxidase, this will be oxidized glutathione, GSSG, and H2O. Okay, and the catalase that is within the peroxisomes works in probably the same way as the glutathione works. Now, the most damaging of all the free radicals is the hydroxyl free radical. And when we form hydroxyl, or when we have a defect of the clearance of superoxide or hydrogen peroxide and so on, basically there would be cell injury. And number 1 is going to be lipid peroxidation. Okay, because that polyunsaturated double layer of lipid can actually get broken down by free radicals, and it could cause membrane damage, and it will lead to the formation of peroxisomes, which are by themselves harmful, and they lead to an autolytic pathway. And of course, protein modifications can occur, and they can actually fragment a couple of protein strands, or break down or misfold proteins. And of course, DNA damage, because especially with thiamine, it could cause a single strand break within that DNA, and it could cause mutations. So actually, the thing is, I forgot to mention that 1, 2, 3, and 4 antioxidants that we take from the vitamins. Okay. Now you hear, of course, in the market, this is an antioxidant and it could help with aging and reduce the risk of cancer. And basically, how that works is by basically preventing the DNA damage, because it prevents the formation of reactive oxygen species and prevents the DNA damage, and so it prevents mutations, okay, or it prevents the protein breakdown, so it helps with aging and with the prevention of cancer. Okay, so actually, what are the pathological causes of free radicals? So actually, there are a couple of causes that I want to mention, and the first is being radiation. And so basically, in radiation, what happens is that the ionizing radiation actually hits water within the tissues, and so it breaks down water into a hydroxyl free radical. Okay, and so when it comes to radiation, it is dangerous because it forms the hydroxyl free radical, which is the most dangerous one. Toxins and reperfusion injuries and inflammation can also cause free radical formation. Now let's talk about the inflammation. Of course, this we talked about this right here. This is a let's say a lysosome. I'm sorry, a leukocyte. So this is a leukocyte, and it has a phagosome, okay, or a phagolysosome. And that phagosome has an enzyme that is called NADPH oxidase. And this is a really important enzyme because it turns oxygen into superoxide, okay? And then by superoxide, by myeloperoxidase enzyme, will turn into hyperchloride. And it will bleach the microbes via this pathway, okay, via the formation of hypochlorite, and by turning the oxygen into superoxide and the superoxide into hypochlorite. And this is a defense mechanism that leukocytes actually use in order to bleach out or kill microbes. Now another thing is metals. Now metals such as iron or such as copper, when they accumulate, of course, why do we have transferrin and ceruloplasmin? Because the iron and copper, they cannot be left alone within the blood because they will form free radicals. So here, this is something that is called the Fenton reaction. And this Fenton reaction, basically, of course, we said that the superoxide dismutase turns into hydrogen peroxide. And hydrogen peroxide, when there is an excessive iron or copper or a metal, it will turn into a hydroxyl via the Fenton reaction. And this is why we get tissue damage in diseases such as Wilson's disease, which is an excessive accumulation of copper, or hemochromatosis, which is an excessive accumulation of iron. We will continue in the next video.